Hello everyone. Hope we had a good Thanksgiving. Today we're going to do this short video first, and then there's a longer one. You'll probably get this one, the longer one, later tonight. But let's begin. This lesson concerns this idea of something called the central dogma of molecular biology, which is sort of the central controlling idea that we always come back to sort of when we're looking at molecular biology, sort of the main concepts or important sort of process that involves molecular biology or DNA. So first of all, you have heard this term of a gene, even if you're like a regular person. And a gene, the direct sort of definition for a gene, is a segment of DNA that codes for a protein. There are many parts of the DNA that control the gene or actually don't do anything, but a gene particularly is the segment of a DNA that has the instructions to make a protein. This PowerPoint says we have about 20,000 genes. There are more, about 30,000 genes we have in our DNA that have instructions to make a protein. This is only about 2% of our total DNA. The other 98%, which either has no function or has actually really important function of controlling sort of how our DNA is controlled and when, or controls when certain DNA genes are turned into protein and when they're not. Just to quickly review about proteins, and I'm not at school, so my writing is going to be bad because I'm doing this using the pointer. So proteins are made, if you remember, of amino acids, and there are 20 different ones. If you remember correctly, they bond together through something called a peptide bond between the C and the N. What the DNA does, and I can't draw a peptide very quickly. Peptide, you could fill in bond. What the DNA does is the DNA codes for the specific amino acid that's going to be inserted into the protein to bond together through a peptide bond. It also, remember that these proteins have different characteristics. They can be polar, non-polar, acidic, and basic. And remember, the acidic, basic, and polar ones, they're considered to be hydrophilic, or they like water, and then the non-polar ones are considered to be hydrophobic. And the other thing that's important about proteins, which we spent a lot of time on, is that proteins fold, if you remember, to form three-dimensional shapes. And that three-dimensional shape determines its ability to function or not. So on this slide, I would make sure you've written a summary of the sort of what we reviewed here about proteins. So how does DNA code for our cells and bodies? Well, first of all, the DNA or gene is in your nucleus, and it codes for protein. These proteins make up most of all of your cells. Part of almost all of these sort of features of your cells are composed of protein. And then the cells obviously make up our bodies and do the job. There's Ralph. Hi, Ralph! Hope Ralph had a good Thanksgiving. He looked kind of fatter and mucher. So the central dogma of molecular biology has three processes. In the first process, DNA, is replicated, which we've discussed previously with the DNA helicase, the DNA polymerase. But that does not do anything to sort of make a protein. There are two processes that do things to make protein. First, we take DNA in the nucleus and transcribe it to RNA, or also known as mRNA or messenger RNA. And that process is called transcription. Then that RNA will travel to out of the nucleus to the ribosome, where that RNA will be translated into protein. And that protein will produce a trait. But that trait is most of the time a functioning protein that does a job the body needs, but that trait is also sometimes something like blue eyes. But just to review this once again, in the idea of the central dogma, you start with DNA in the nucleus, and the DNA cannot leave the nucleus, so it gets transcribed, or the process of transcription, into a molecule of RNA, or also known as mRNA, and the M stands for messenger. Then that mRNA, once transcribed in the nucleus, travels to the ribosome, where it is translated into protein, and that protein will do its job. Again, most proteins will perform a vital function in the body, and if you don't have that protein, that can lead to a disease. And other proteins also cause visible, seeable traits. Eye color, hair color, height. And then Ralph down here saying that DNA gets all the glory, but proteins do all the work, which is true. The DNA has been sort of glorified as this amazing molecule, but it's the proteins that particularly do all the jobs in our cells. Like I said, a lot of times if you have a non-functioning gene that codes for an important protein or enzyme, that will lead to some sort of disease. So if you look down at the pathway, we have a molecule A which turns into molecule B, C, D, so forth, and all of these processes are catalyzed by enzymes. If one of these enzymes in the pathway has a gene that is non-functional, or a gene that has a mutation, which we'll talk about much later, next week, then that leads to a disease, because probably E in the pathway is really, 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 really important. Some of the sort of common diseases that we have that are associated with a gene that's mutated, or a gene that's not working properly, or doesn't make the correct protein, is sickle cell anemia. And sickle cell anemia is when the hemoglobin protein has the wrong amino acid into it, because there's a mutation in the DNA, which causes that protein to fold wrong. And then it can't carry oxygen efficiently, and it also has a strange shape that causes clots more likely to form. Then we also have a disease called PKU. And in PKU, you can't process the compound phenylketonuria, which is in Diet Coke. You don't have the enzyme to break it down, and not being able to break it down if you ingest that can cause you to be really sick or feel sick. And then last but not least, another disease that we have is I can never say this correctly, kind of like parallel, but albinism is when you can't make melatonin, or the pigment in your skin. People who have this disease, their skin appears extremely, extremely white because they can't make any of the pigment. They also tend to not have pigmented eyes or pigmented hair, so their hair will also be white in color, and their eyes actually usually appear red in color. For these type of people, since melatonin protects you from the sun, going out into the sun is a very, 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 very dangerous sort of proposition, because they have no pigment to protect them from the sun. But the point is, all these diseases here are caused by a gene that has a wrong A, T, G's, and C's. Because the A, T, G's, and C's are wrong, it calls for the wrong amino acid, and then as a result, the protein is not correct, or not the right shape, and then the protein cannot do its job, and if the protein cannot do its job, that leads to a process not occurring correctly in the body. Just to summarize again, here is the central dogma of molecular biology. So the central dogma says we start with DNA in the nucleus, which gets copied into mRNA, and that process is called transcription. And then mRNA leaves the nucleus, travels to the ribosome, and it's turned into a protein. Well, we're going to learn all about this process in the next video. And again, that protein, which it will do a specific job, and then produces a trait, which Ralph was standing under. Two words that are commonly used that are confusing sometimes are the words genotype and phenotype. Phenotype, specifically, is the bases, or the DNA. So that's the DNA bases, or the code that you have in your DNA. The phenotype is what you see. So, for example, with blue eyes is the phenotype, but the A, T, G's, and C's that cause me to have blue eyes are the genotype. The genotype refers to your DNA. The phenotype refers to what you see in the organism. Okay. We're going to be spending now sort of in the next video on transcription and translation. Before we spend a lot of detail on transcription and translation, we have to talk about sort of how mRNA or RNA is different from DNA. Remember, mRNA will give the instructions to the ribosome on how to make protein. Before we talk about that process, we need to talk about how mRNA is different structurally from DNA. So first of all, the sugar is different. DNA has the sugar deoxyribose, that's where the D comes from, and RNA has the sugar ribose. They're different, and I don't know if I have the picture. I don't. They're different in the sugar. If you look at the sugar here, they all have a sugar. They're different in this here. In deoxyribose, this is an H. And then in ribose, it's actually an OH. But that's how they differ. The nitrogen bases are also different. RNA has no T. Instead, RNA has U. So in RNA, T replaces U. Secondly, RNA is single-stranded most of the time. There are some molecules that actually fold upon each other, like RNA has the ability to fold as well, which is kind of like a protein. But in generally, mRNA in particular is single-stranded. And there are also many mRNA molecules that exist where there's going to be sort of only one DNA molecule in the nucleus. There's tRNA, which we're going to talk about in the next video. Then there's mRNA, which we just talked about. And then there's rRNA, which actually makes up the ribosome. So there are many different types of RNA molecules, where there's only one type of DNA molecule. So here, this is the difference. So these show the differences. There's the sugar. So here we have ribose versus deoxyribose, and we see the only difference between ribose and deoxyribose is that here there's an OH in the ribose, and in deoxyribose there's U. Here we also see the difference in bases. We have U replacing T. And then last but not least, we see here that DNA is double-stranded, and we also see its anti-parallel structure. The one line running 5' to 3', and the other running 3' to 5' with the phosphate group down. And then we have RNA, which is only single-stranded. So therefore it can't be anti-parallel, because it can only has one strand, it can only run 5' to 3'. Alright, well, stay tuned for the next video. There's going to be a little bit of delay between the next video and now, because it's time for dinner. But the next video will be released sometime tonight. I will see you guys on Monday.